So most of you probably already are familiar with principal component analysis, if you're already doing any data analysis in general or bulk RNA sequencing. So it's a linear algebraic method for dimensionality reduction, and it's a case within the singular value decomposition method, which is very used for data compression. And it states that any matrix can be decomposed as a multiplication of other matrices. So that's the term factor of matrix factorization. But we can illustrate this as a multiplication of matrices like this. So this is the original matrix that you have your genes and your samples. And that matrix can be a multiplication of a genes versus principal components versus another matrix of samples times samples and another matrix of samples times principal components. Being these principal components are, so like the idea of PCA is to find which values for those three matrices can be multiplied together to generate your additional matrix. And basically this matrix here does not exist in normal PCA because you're scaling and doing the Z transformation that also mentioned. So if you just look at the normal principal component function in R, you'll have, for example, the matrix rotation here and the X. The center and scale are actually this matrix here. And this indicates for these principal components, this reduced space, which I'll come in the next slide, what is the importance of each sample to each principal component, and what is the importance of each gene to these principal components. So how does it work visually? So here, imagine that you have two genes, A and B, and then you see that they are quite correlated to each other in this case. So the information that gene A and B convey is exactly the same. So if A is high, B is also high. So here you have your data in the Z-score space, and then you pass through a line over the most variance part of your data. So like the red line in here. The same thing goes with the blue line, which will be 90 degrees of the red line. And if you have many other genes, you'll just put another line, also 90 degrees to both of them. So if you find the variance all over those points, so you can see that this is where you have the most variance in the data because all the data is spent in this direction. And that will correspond to principal component 1. The same way, 90 degrees to this principal component 1, you define another line that spans the most variation that you can get, and that will be principal component 2. That spans most of the variation in this direction. So, and this is how the PCA will look for this data set. And basically, PCA is actually just a rotation to another coordinate system, where principal component 1 will be where in the line most of the variation in your data set can be captured, and principal component 2 will be the second most variation that from your data set that can be captured in this coordinate. And from this plot here, we can also see that most of the variation that we see in principal component 1, which will be the sum of all these distances here squared, when you can just sum all of them and see that most of the variation is stored in principal component 1, and just a tiny bit of information is stored in principal component 2. And that gives you an indication like how much information is stored in each one. For then we can conclude that PC1 explains 98% of the variance. And thus, principal component 1 can represent those two genes very well. Since they are quite correlated, I don't need two genes to express that behavior. If A is high, B is also high. I can just use PC1 as an illustration of that expression pattern. And that way we remove redundancy of two genes that are representing the same idea. PC2 in this case is nearly insignificant because it stores very little information. So we can just ignore that one. And that will be basically what we would consider noise in the data. And in real life, what we see for single cell, we don't see like a very clear trend like this, but we see an exponential decrease in the variance. And in that case, we can use, for example, the top 50 principal components, because those are the ones that store most of the information. Because after, for example, after 50 here, you see that there is not much information change between the different principal components. So it's a common use is to define your principal components and then set a cutoff on how many principal components you actually want. Another cutoff that you can set is how much variation is stored in the principal component. But the common one is usually a number of principal components that you choose. In single cell data, you will see that there are only a few data sets that can be visualized using principal component, especially if they are not of super high complexity. So in here, you have two different data sets. Here you can see the different cell types. In this one, actually, there are I haven't colored the cell types in here, but you cannot usually see. But it's important to know that principal components, they are usually correlated to the sequencing depth and the heterogeneity of your cells. Not always, for example, in this case, because the cells are very similar to each other. But if you have different cell types, usually what you'll see is that the principal components will also correlate. If, for example, red blood cells will be clearly separated because they have very low amount of RNA, or cells that are very complex. So, for example, epithelial cells, they also will be very well separated because of this complexity, not because of the marker genes. And as I mentioned before, you have those two matrices that are multiplying together. One of the matrices will correspond how much each sample is important to each principal component. And in the other matrix, you'll have how much each gene contributes to each principal component. And in that case, you can find information about which genes are important for driving the separation in principal component 1. And that's also what also mentioned before, that you can use these genes, for example, to select and further do clustering analysis if you want. So to keep in mind, PCA is a linear method for dimensionality reduction. It's an interpretable dimensionality reduction because the principal components have a meaning. So principal component 1, the highest variation. Principal component 2, the second highest variation, and so on and so forth. As well as we know which genes contribute to each principal component separately. So that gives an interpretation of the dimensions. The data is usually scaled prior to PCA using z-score or using the scale data, for example, in Seurat, or the VAR transformation in SCAN. For the top principal components, usually contain the highest variance, as I mentioned. And then you can use that for filtering. So use the PCs that explain 1% of the variance, use Jackstraw to test if that principal component is actually more significant than a null model. Seurat has also this implementation, but it's very slow for big data sets. Or you can also just select how many principal components you want. So here it's just an example, 5 and 10, but this is very, very low. So I would recommend 20 to 50 or more. But the problem is that it performs poorly on separated zero inflated data because PCA is linear and single cell data is very well, is quite big and zero inflated. And in that case, that's why PCA is not very useful to separate or visualize your data in that space. And the cell size and sequencing depth are usually captured in the top principal component. So you need to pay attention to that as well. Not that you need to regress that out, you need to include that information as well. But just so you know that these are commonly seen in single cell.